transcription has started. That's good. And recording has started. Great. So it's about 4 o'clock now, Sam, and you are the other side of London, right?
Indeed, I'm at the Crick.
At the Crick. So you could have walked over and given a seminar, but we're still doing these online. And the benefit of that for our seminars being online actually is I think quite a few people who are dropping in are in the middle of maybe working in the lab or something, and they've got an iPad out on the bench. They're going to watch you and hear about your research. Other people will be at home. Some people may be in other countries. And like Kemi said, this is being recorded, so this will also be on YouTube, and some people can watch later.
Great.
And email you and pester you for positions in your group, hopefully.
That would be awesome.
Yeah.
That would be fantastic.
But it's we will get you over in person sometime soon, because there's lots of good possibilities for connections between your work at the Crick and our work in the bioengineering department at Imperial.
Fantastic, yeah.
So you're going to talk to us today for about 45 minutes or so, and then we'll have like about a 15-minute formal period of questions. And then if people are able to afterwards, from 5 onwards, we can stay on the line and chat and catch up. Is that cool? So we were trying out your slides a minute ago before everyone came. We have the quirk with the Teams system today. There's always something wrong with Teams. That when Sam shares his slides, you can no longer see his face. The webcam goes off. So that's why we don't have the slide deck up yet, so that you can all see what Sam looks like. But the moment he starts sharing, you're just going to get the joy of seeing his slides as well. So I won't give any spoilers about what he's going to talk about today. He said he's been umming and ahhing about what to talk about for quite a while today, and so there's no fixed agenda. We'll just see what he wants to share with us. But let me just tell you all that Sam is a recent recruit at the Crick since January 2021, having previously been a postdoc in MIT, partly in the media lab and partly doing research in brain cognitive function. But predominantly, the thing you want to know about Sam is he comes from a physics background, did physics as an undergrad, spent some time at Cambridge as a master's student and as a visiting student, a scholar as well, so he's familiar with the country. And he really is focused on technologies, tech development, and particularly, like, you know, how that can be applied and turned into companies and technologies that can be shared and commercialized as well. So there's a particular back history of working with venture funds, considering other models other than academia for doing research as well. I don't know if you're going to get a chance to tell us anything about that today, are you?
I hadn't even thought about that, but I can like throw in. Now you're trying to
discuss that a bit later. These interesting things like focused research organizations and stuff like that are coming in. Can you promise me one thing though?
Yeah.
Don't say Petri, say Petri. I hate it when Americans say Petri dish.
I cannot promise that I will not say Petri. I'm sorry.
Okay. All right.
Yeah.
The floor's yours now. So feel free to start saying hello to everyone and sharing your slides, and I'll shut up and stop taking so much time.
Great, thank you. Yeah, hey everyone. So that was a fantastic actually introduction. That was one of the more on-point introductions that I've gotten. Actually, so really, Tom, thank you so much for that. You really kind of hit the nail on the head. I will say a couple of things before starting. The first thing is that so I can't see anyone right now, and I gather that you guys can't see me. So like, really, please do feel free to jump in, because otherwise I'm just sitting in my office talking at a screen, which is like, you know, less fun than talking to actual humans. So like, really, if you have like a question you want to interject, just like jump in, and that would be totally fine. The other thing is to plug that, yeah, indeed. So I'm an inventor. Basically, I'm an engineer, and I'm going to tell you today about some of the things that I invent, some of the, like, the biotechnologies that I invent. But I also invent new ways of funding science, which is a little bit meta. And one of the thing that Tom was referring to is that one of the new ways of funding science that I have invented is called a focused research organization. And it is basically a nonprofit that is intended to take on a project that is too large for an individual lab, but that doesn't have an immediate profit motive, and so it can't be funded by like venture capitalists, or it can't be funded by investors. And we came up with a specific structure for funding those projects, and we have 3 of those projects that have just been announced. They were announced on October 12th and 13th, which are being funded at the level of $50 million each by 2 different philanthropic funders in the US. And one of the other people who's working on setting up those organizations is going to be at the Crick on Friday at 5:45 p.m. To talk to people at the Crick or from the broader community about what focused research organizations are, and they're looking for more proposals, and they're looking to fund more of them. So I think if you have good ideas about projects that are just like a little bit too big for an individual lab, but that are also like not really oriented towards short-term profits, or if you're interested in other career paths outside of academia, or if you're kind of just interested in like philanthropic funding and that kind of process in general, shoot me an email. My email will be on the slide at some point, and I'd be happy to invite you to this event on Friday evening. It'll be very cool, and you can get to hear at that event a little bit more about these ideas of how we can accelerate science through new funding structures. Otherwise, I also just like to start out by saying that I am half scientist and half entrepreneur, and so I have that generates some potential conflicts of interest. But I just like to disclose up front that I'm a co-founder on 3 companies, one of which called Helios is commercializing a device for drug delivery to the intestines. One called Siren 5 is a neurosurgery robot, and then also a reagents company, which is still in stealth, and then I have informal consulting relationships with Wobble Genomics, recently spun out of the University of Edinburgh, and Irradiant Technologies, which is commercializing implosion fabrication, which is another technology that I invented. It's a nanofabrication technology I invented in my PhD. So, and then the last thing before we like really dive in is that I am recruiting people. I just got several grants in the past few weeks, and so I have positions for a bunch of postdocs. And so if you're interested in the kinds of things I talk about today in biotechnologies, and in particular inventing biotechnologies that can be deployed widely through like commercialization and getting those technologies like beyond just like a high impact publication in a journal, please reach out to me. There's my email, and this I was saying before, if you're interested in coming to the FRO event also, there's my email, and you should email me. I love to hear from people. So okay, so the way that I like to describe what we do is that I say that we do blue skies bioengineering with high impact applications, right? So there are a lot of labs in the world that do kind of blue skies bioengineering, working on like figuring out how fundamentally to do new things with biology. But I think that the thing that really sets us apart is that a lot of the labs that do this kind of blue skies bioengineering end up missing out on the impact of the technologies that they invent because they don't have the actual pathway all the way through to deployment like work at. And so a lot of what we focus on are finding problems and finding ideas that are both like really satisfying and really fun and exciting to work on from like a basic technological perspective, but that also have like a clear path to actually getting out and helping people, right? And if you think about the actual pathway to get from like a problem to like a product or something that can help people, it is long and full of terrors. And for example, you can have a problem, and like often the problems that people work on in academia just like aren't actually that important at like a broader societal level, right? Like or maybe you have an idea which doesn't work. I mean, I think we all here have a lot of experience with that failure mode. You publish a paper, but then you don't get it spun out into a company. You publish or you spin your idea out into a company, but something is wrong with the way the company is set up, or like for one reason or another the company fails, right? And there are all those failure modes that you can run into before you get to a product. And so what we like to do is when we're thinking of a new project, we like to think about, for all of these possible failure modes, how do we go about minimizing the likelihood that we end up in one of them? right? So from the problem, like the problems that we work on in this lab are all ones where we can, almost all ones where we can like validate from the beginning that this is a problem that like if we solve it, there will be people lined up to use it, right? Which is really way more difficult to validate than it sounds. That's actually the hardest part, right? We have ideas, they don't always work. There's not that much we can do about that one, right? But like then we also make sure that we have like relationships with investors and we have relationships with entrepreneurs so that when we're getting close to the point of wanting to publish a paper, we can start to have conversations about how we're going to get that technology out the door, right? And just like get all that stuff rolling early so that we like get as quickly as possible from the idea of the publication to something that people can actually use, right? And then also building those relationships with like people who are really experienced in company creation, really experienced in investing, will maximize the likelihood that eventual company can be successful. And so these are the areas that we actually focus on. And for the most part, these are centered around, well, mostly like medicine applications, and then like fundamental and like kind of like applications in accelerating science, right? So we have a bunch of projects that we're working on around drug delivery. So figuring out, I like to say, figuring out how to deliver any drug to any cell, right? So viral vectors and LNPs, how do we deliver nucleic acids, or how do we deliver biologics to specific cells? ADCs, like antibody drug conjugates, like kind of next generation versions of ADCs, more generally, like how do you get small molecules to cells in a very specific way? And then, so that's like the first category. The second category is diagnostics, which is like, how can we diagnose any disease 15 years before when it happens, is the big overarching goal there. And then omics is really like, how can we visualize like the properties of biological tissue in their spatial and temporal, in their native context, right? So obviously biology has been transformed by the emergence of single cell RNA sequencing, and but single cell RNA sequencing requires you to actually go in and dissociate the tissue before you can do the sequencing. And in the process, you lose the spatial information, you lose the temporal information. In the context of neuroscience, you lose information about where those cells are in the circuit. And so, that kind of branch of the lab is focused on being able to measure properties of biological systems in their context, right, in their native context. And so what I'm going to talk about today, primarily, so because we care a lot about, commercialization and deployment, we also care a lot about intellectual property, which means that there are a lot of things on here that I can't talk about. So actually most of the lab, the majority of the lab is working on this drug delivery side. And there's almost nothing that I can say about that. We have a patent which is winding its way through, and I was hoping it would be submitted by now, but it looks like it's going to be another week or so. So at the end, I'll probably mention something about what we're doing in that area. Otherwise, I'm going to focus today mostly on temporal transcriptomics and connectomics, which are the two areas where we have where we've either published or where things are furthest along, because those are kind of continuations of the work that I did before. And then I'll also hopefully have time to mention something about some of the diagnostics work that we're doing as well. So that will be kind of the outline. So to start with, temporal transcriptomics and connectomics. I should also mention, so the thing that, I also did this like spatial transcriptomics thing called Slideseek, which is basically single cell RNA sequencing with spatial resolution. I feel like I just talk about that a lot. And so if you're interested in that, feel free to get in touch or anything like that. But I thought that I would change it up today and talk about some of the other projects that we're working on going forward. So to start out with is temporal transcriptomics, a method which we call timestamps. This was work that I did while still in my PhD with Fei Chen and Ed Boyden and Linlin Chen. So Ed is in the top left, Fei is on the right, and Linlin is in the bottom left. And Linlin is my co-first author, who's absolutely brilliant. She co-first authored this Nature Biotech paper as an undergrad. and then she's now is like in her second or third year of grad school at Caltech. So I keep messaging her to ask her when she's going to come and do a postdoc with me. But anyway, I can't speak highly enough of them. The fundamental, so again, like the idea here is that RNA sequencing is basically an endpoint measurement. So it lacks temporal resolution, right? And so some people have tried to come up with things like RNA velocity in order to restore temporal resolution to RNA sequencing. That provides you with like an instantaneous derivative, right? Or like there's Slamseek, which allows you to go back and say, what are all the RNAs that were created after a specific point in time? So there are methods that people have been inventing, but our question was really like, how do we actually create like an image of the, so to speak, of like the gene expression over time? Like actually look back in the past and say, how much of this RNA was created at each point in the past, right? To say like, oh, like this RNA was created 10 hours ago, then like there was some more of it created 5 hours and some more of it created 2 hours, right? So we can really just, in the same way that like we can add a full spatial dimension to RNA sequencing using things like Slideseek or Visium or, Merfish, or name your favorite method. The question is like, how are we going to be able to go back and add a full temporal dimension to RNA sequencing? And the central concept, if you remember nothing else about timestamps, the central concept that you have to remember is that asking when a transcript was created is the same as asking how old that transcript is, right? And so if you want to figure out like how, what the cell was doing 10 hours ago, you ask what are the transcripts that are currently in the cell that are 10 hours old. And so basically what we then did is that in order to add a temporal dimension to RNA sequencing, we went and we created a way of measuring the age of RNA transcripts, which is hence the name of the paper. RNA timestamps identify the age of single molecules in RNA sequencing, not because we're particularly interested in the age of RNAs, but because we want to know what was happening at the time when they were made, okay? And so the way that it works essentially is that we use RNA editing. So there are these enzymes called ADARs, adenosine deaminase, acting on RNA. ADARs basically convert adenosines into inosines, which are then interpreted by reverse transcriptases as guanosine. And so if you have an ADAR that's acting on your RNA, you see all these cases in which like an A has been converted into a G, right? And basically what we just said is that we can build a cassette we can build like a specific RNA cassette that has a bunch of adenosines in it, attach ADAR to the RNA, and then ADAR will progressively over time like edit these adenosines so that they turn them into inosine. And by counting the number of inosines, we can like figure out how old the RNA is, right? It's like seems straightforward, and turns out it is straightforward. Those two things are not always the same. There are lots of things that seem straightforward that are really, really hard. But in this case, we gave it a try and it kind of worked. And so this is actually an image of what this cassette looks like. So basically what we did was that we took like a gene that we're interested in. So for most of the for most of what we did in the first paper, that gene was just like RFP. So it's just like a fluorescent protein. And we added this thing, which is very highly double-stranded because ADAR typically likes to edit double-stranded templates, and has these MS2 loops so that you can stick ADAR onto it. We took ADAR, we replaced ADAR's endogenous RNA binding domain with an MCP binding domain, which binds to MS2 and which also then decreases off-target effects. So you can express ADAR in cells without like, messing up other transcripts. And then if you look over time, so basically like the structure of these experiments is that we have this time-stamped plasmid, this time-stamped RNA expressed under a doxycycline-dependent promoter. So you add doxycycline to the cells for an hour. You then use actinomycin D, which just blocks transcription to stop them. So you generate a pulse of transcription, square wave, and you lyse the cells at different points in time after actinomycin D, and you look at like what do the RNAs look like, and what you see is that just like qualitatively, just like from these 4 randomly chosen reads, as time goes on, there are like more there's a greater incidence of adenosines being edited to guanosine. And it's actually like really highly reproducible. So on the right here, if you look at like, how many edits there are per transcript at each of these time points, these error bars are across like biological replicates. So it's like, it's really very highly reproducible. It gets fit extremely well by an exponential CDF, which is what you would expect for a Poisson process, which is what this is, just like adenosines being edited randomly in time. So then you can go in and you can say for a transcript with a given number of edits, what does the distribution look like over the possible ages of that transcript? And that's the graph that I'm showing you here. So you can go over and you can say, oh, if my transcript has 30 edits, can I infer how old that transcript is? And the answer is, yeah, that transcript is probably 7 hours old. It could be 6 or it could be 8. And basically, it turns out that we can localize the age of an individual RNA with approximately like 2.5 or 3 hour time resolution, which is pretty cool over 12 hours. Now, that in most cases, you don't have like a single transcript being expressed. You get a lot of transcripts being expressed together. And so then you can ask, like, if you have an ensemble of transcripts, how do you go about figuring out what the underlying transcriptional program is that gave rise to that ensemble, right? And for that, we built basically an algorithm. The way this algorithm works conceptually is that we know what RNAs look like that are 1 hour old. We know what RNAs look like that are 2 hours old. We know what RNAs look like that are 3 hours old, and so on. And so for a arbitrary distribution of the number of edits per RNA, right, we can ask what is the transcriptional program that is most likely to have given rise to this distribution, right? And then conceptually, the way that experiments work with this kind of a system is that you start out with some underlying transcriptional program, which is over here on the left. So for example, your ground truth program might be, it's just like there was a burst of transcription 2 hours ago and then no other transcription. That underlying transcriptional program gives rise to a underlying RNA distribution, which is like over here. So this is like a distribution of the number of edits per RNA, right? And that's like the ground truth. Now in the experiment, you get some sample of that distribution because like cells don't have an infinite number of RNAs in them. They have a finite number of RNAs. So you get a sample, which might look like this thing over here on the right, which is like a very poor approximation to that underlying distribution. And then from that, you go and you use this maximum likelihood algorithm, which is going to give you an estimate based on this like observed RNA distribution of what the transcriptional program looked like. And in this case, this is like a real example, right? In this case, it estimates that it's like half 2 hours and half 3 hours with like a little bit of weight at 1 hour. And that's actually like pretty, I mean, given that this is an example where there are only like 20 RNAs sampled, I think that's like a pretty good estimation given this the like ground truth transcriptional program, right? So once you have this algorithm, you can then go and you can apply it to kind of complex scenarios. So we in this experiment, basically we took time-stamped RNAs being expressed under 2 different promoters, a doxycycline-dependent promoter and a light-inducible promoter. And we had one of them induced at I think 2 hours and 1 hour, and one of them induced at 5 hours, right? And that generates this distribution of edits per RNA that has 2 humps, right? Because there's a burst of transcription, those RNAs start aging, and then you have another burst of transcription, which generates newer RNAs, and you get this like 2-peaked distribution, which is what you see at the top. We then went through and downsampled it to kind of simulate what you would get if you were actually looking at like an individual cell. And we then applied our algorithm, and you can in fact pick out, even with like only 50 RNAs in the cell, you can in fact reliably pick out that there were 2 bursts of transcription at different time points, right? And again, you get this kind of thing where you're like not sure if it was 1 hour ago or 2 hours ago, but the fact that you can like really clearly identify that it's 2 separate events is pretty cool. And so that was like simulating what it would look like in single cells. So we then went and actually applied it in single cells. You know, this is like relatively straightforward. Basically, we had cells that were induced with doxycycline like 1 hour ago, 2 hours ago, 4 hours ago, and then look at the distribution of the edits per RNA. And that's what this heat map is showing you. In this heat map, every row is a cell. We're just showing you how many edits there are per RNA in each of those cells. And you can then go and do some clustering, and you can predict based on your clustering whether the cell that you're looking at was one of the 1 hour, 2 hour, or 4 hour cells. And you get it right with about 90% accuracy, 85% accuracy. So that's pretty cool. Okay, so that's the story for timestamps. We're working, so in the lab, we're working actively on a few different methods of extending this to the entire transcriptome. I am not going to say any more about that right now, but if you're excited about temporal RNA sequencing and want to think about ways of extending it to like get temporal resolution across the entire transcriptome, we just got a grant for that from the Impetus, from one of these Impetus grants for aging research. The idea is to use transcriptome-wide timestamps to look at the processes that lead to senescence. And so if you're interested in aging or in temporal sequencing, please reach out because I'm hiring. Okay. The second story to share is about connectomics. So connectomics and temporal transcriptomics, they might sound pretty different. I think that they are not that different because, again, the question here is, how do you go about looking at like elements of RNA, like gene expression, like in the context of networks, right? Like in the context of neuronal networks, instead of having to like dissociate those neurons and lose all the information about the connectivity, can we figure out how to get that molecular information out, like in network context? And you know, basically, we know that the brain is characterized by like extraordinarily non-local interactions, right? Like, for example, this is like for the mouse light data set from Genelia, and you know, the average, I think the average like process length per neuron was like 1.3 centimeters or something. Maybe it was 10 centimeters. Now I can't remember anymore. But the point is, it was on the order of centimeters, which in a mouse brain should really tell you that this is like highly non-local. And so this idea that like, you know, everyone wants spatial transcriptomics because it allows you to see like what cells interact with each other. That works great everywhere except for the brain. And in the brain, you can't do spatial transcriptomics. You know, the spatial transcriptomics is not going to tell you about what neurons are interacting with each other, right? You need the connectomic information. And so connectomics is traditionally mapped using electron microscopy. Basically, you like take the brain, you stain it, you slice it up into tiny sections, like 60 nanometer sections, and then you image them. And then you can like reconstruct from those sections what the circuit looked like. And there's been a ton of progress here, but it completely discards any molecular information, right? And people have been trying to figure out how they can incorporate molecular information into connectomics, into electron microscopy connectomics, but I think that it's a long way away. And so what we wanted to do was come up with a different paradigm that would enable connectomics with molecular information. I kind of already said this, so I'll just say it again. I think that there's like a analogy to be made here between you know, genomics and connectomics in that you know, the genome, like first we sequence the genome and then we realize that the actual sequence of the primary sequence of the genome only goes so far. And so we realize that like actually you need this thing called like epigenetics, which tells you about like the function of the genome, right? Which is essentially looking at what are the proteins that are present and like what are the functions that they mediate. Connectomics is like the same thing, right? Like the process, the electron microscopy connectomics that people are doing right now, which like, you know, this audience may not be tapped into neuroscience. So I will say like the Allen Institute recently published a full cubic millimeter of mouse cortex, which is like a huge leap forward. It's like, 10 times larger than anything anyone had done before. And it's, 1,000 times larger than what we had 5 years ago, right? So like progress is being made extremely rapidly in the area of connectomics. But again, we're at this stage of like, we're at the stage of just looking at the primary structure of the connectome, and we're missing out on all the molecules that provide function, like, synapse stains. And so in the same way that we have like, epigenetics, I think we should probably be thinking about like epiconnectomics, basically, not mapping not just the connectome, but also what are the proteins on the connectome that actually give the brain function, right? So that's the broad vision. And so the system that we're building has basically 3 components: super resolution microscopy, multiplexing, which is a way of visualizing many proteins or RNAs at a time, and then molecular barcodes. I'll just describe each of these kind of quickly. The first component, super resolution microscopy. So when it comes to visualizing, the functional proteins in the brain, which are like mostly located at synapses, historically, the problem has just been that synapses are super small. And so it's really, really difficult to actually visualize them. But in, one of the advances of the past 5 or 10 years has been the emergence of super resolution microscopy methods like STORM, or pictured here as like expansion microscopy, which now make it really easy, really straightforward to visualize synapses. So like on the left here, for example, these are dendrites from a 20x expanded brain section, where like those dots that you see are actually like individual molecules of GFP. And then on the right, this is a 4x expanded brain section, but still plenty high resolution to see the dendrites. And these things in blue and pink are Bassoon and Homer, respectively. They're pre and postsynaptic stains that show you where the synapses are. And so the idea is that like the kind of images that we're looking for are images like these, and it's now pretty easy to go and get them using these super resolution microscopy methods. The second component is highly multiplexed RNA and protein imaging. If we want to look at the function of neurons and the function of synapses, there are a lot of proteins that mediate those functions. Typically, it's actually like not even that easy to get information about those functions out of transcriptomic data, which is part of why I'm not talking about like sequencing right now, because really, just because a neuron is expressing some particular transcript doesn't mean that it's like at a specific synapse. And what you really want here is like to get information about like individual synapses, right? And so for that, we need to go to imaging. And another one of the advances in the past 10 years or so has been the emergence of new techniques for like measuring hundreds or thousands of RNAs or proteins in the same sample. So this is just an example from 2 years ago, which already feels super old in this field, of like imaging 12 different markers in the retina at the same time. Commercial systems are coming out that can do hundreds. And so this is like a major area of improvement that will be very enabling for connectomics. And the third component is barcoding, right? So I started out by saying that interactions in the brain are extremely highly non-local. And so you don't want to have to go and trace all 10 centimeters of, if you want to like measure an interaction between a neuron in region A and a neuron in region B, you would really prefer not to have to go and like track the entire axon from region A to region B, especially because like often the axons like start out in like, the cortex, and they will like wind all the way around the brain, right? And so if you actually have to go and in electron microscopy style, like image a single neuron that image the projection of a neuron from region A to region B, you're going to basically have to map the entire brain and reconstruct the entire brain. And so that's not like very tractable. And so the idea is like, how can we go and create a barcoding system that is sufficient diversity that we can actually visualize, that we can identify like a neuron in region A and the same neuron in region B, or a process of the same neuron in region B, without having to go through the entire intervening volume. And I will preempt a question by saying that the reason that, so I'm going to be talking, like, first thing you might think about is RNA barcodes. And indeed, some people have used RNA barcodes. They end up, like, RNAs don't traffic that well in the processes. They end up super sparse. And so we needed, like, you know, there will be like 3 barcodes in region B, and it just is like not enough for identifying the neurons. And so we need a different strategy. And so basically putting all of these, and so I can say actually at this point, like the strategy that we're taking is a protein barcoding strategy, where similar to Brainbow, which is what's shown here, where, you know, you have a number of protein. So in Brainbow, basically, like, every neuron is going to randomly express a subset of 3 different fluorescent proteins, and that's why the neurons in this image all appear a different color. That's like actually what you see under the microscope, because you have 3 different fluorescent proteins, and if you randomly express one of, you know, randomly express each fluorescent protein, there are 2 to the third, so 8 possible combinations, and that's why you can, like, tell this green neuron over here apart really well from, like, this blue neuron or this, like, red neuron, right? But, like, 8 combinations is not sufficient to uniquely identify neurons anywhere in the brain. And so our fundamental idea around barcoding was to say that we can extend this from, like, 3 different barcode proteins to 30 different barcode proteins, so that instead of having, you know, 2 to the third possible combinations, which is 8, you can have 2 to the 30th, which is 1 billion. And then that will be enough to actually give a unique barcode to every neuron in the mouse brain. And so putting these all together, we call this, like, XCOM, so expansion and connectomics with multiplex protein mapping. So the way it works is that you start out and you have these barcodes. You use viruses to deliver these barcodes, or you can make it transgenic. It's complicated. We can talk about it. But you use these viruses to deliver barcodes to the neurons randomly. So each neuron gets a different barcode. You then go in and you expand the sample. You stain the sample and you expand it. And then you can go and you can image each of your barcode proteins in that sample to identify neuron identity. And then you can also go in and, with, like, the same microscope and the same process, you can image these, like, proteins in situ using multiplex protein staining, which will allow you to identify the functions of those processes, so you get both the structure and the function. And so when we were looking at this, we said, well, many of these things, you know, expansion and the protein mapping are largely worked out. The element here that has not been worked out is the actual spectral barcode, and so that's what we've been developing. And we went and we asked, like, how do you expand from, like, 3 different barcode proteins up to 30 different barcode proteins? And the answer for us was that we have been using these things called spaghetti monster fluorescent proteins, which are basically just like GFP scaffolds with different peptide epitopes on them that you can stain reliably. And they're super bright because they have many copies of the epitope on each protein. And so we went and we screened. This is with Bobayan, who's a postdoc in Ed Boyden's lab. We screened, like, 80 different of these protein scaffolds that all can be stained independently, and identified 17 here that work really well in vivo. We're working on extending that to even more. But this is just an example of what some data looks like when what like high dimensional barcode data looks like, which is that these are 2 images of the same region of the brain. They're both 4-color images, and they are different. They're all different colors. So this is like an 8-color Brainbow. There's no way to depict this. If I were to put these 2 images together, it would just all be white, right? But you can see that, like, you know, the neuron over here is, like, pink, and the same neuron over here is blue. And yeah, we're still working on figuring out exactly how to visualize it. But the point is that this method, like having these 8 channels of Brainbow, allows us to do really robust reconstructions of like pretty densely labeled brain, which is what you're seeing. And so then, just to give you an example of what this will look like, this is a volume that we've been working on. This is actually even only 3-color Brainbow, so we're still working on reconstructing. We're working on reconstructing this volume. But even with the 3-color Brainbow, what we've done is that we have gone and built a neural network that is and train the neural network to reconstruct, to use this barcoding information to reconstruct neuronal morphology. And so we're able to like come up with these really high quality kind of EM style reconstructions of like axons on the left, of synapses with like, synapse, like automated synapse annotation on the bottom, different kinds of dendrites and different kinds of neurons on the right. And so, yeah, this is still very much a work in progress, but it's getting there. It is, yep, it's coming along. Let me see, how much time do we have? We have about 20 minutes, and we want to make sure we have enough time for questions. So, yeah, I can give you guys, I definitely want to talk some about some of the newer projects we're working on, because some of the newer projects are the projects that are more applied. I mean, these projects are both things that I started while I was still in grad school. So just to give you guys a sense, I'll run through this pretty quickly. So this is on the kind of more diagnostics side of the lab. And the idea is, just basically, we wanted to figure out how do we detect proteins in a solution at like extremely low, like extremely high sensitivity, right? So basically, for diagnosing things like neurodegeneration, and it's already known that there are protein markers of neurodegeneration that begin to show up like even 15 years before the onset of symptoms, mostly in the CSF, in the cerebral spinal fluid. But it's like very invasive to get a CSF assay. And so a lot of people have been interested in figuring out how do we measure these, how can we measure these proteins in the blood. And so there have been a lot of advances in this recently, like Quanterix has come out with its Simoa system, which is like a digital ELISA, which has extremely high sensitivity. What we started asking about is like, would there be a way for us to convert this protein measurement problem to a DNA measurement problem in order to allow us to do very high sensitivity detection, because DNA detection can be done very very sensitively. And so the idea, which is called Arsenal, which so I should also say the postdoc who was working on this is his name is Patrick Arnott. He came up with this name Arsenal, which when he came up with it, I did not realize that was the name of a football team. So that's then I learned. So anyway, Arsenal, aptamer, release strand enhanced nucleic acid amplification in liquids, definitely a backronym, is basically a way that we came up with to use aptamers to turn protein measurement into DNA measurement, which obviously the idea of using aptamers to turn protein measurement into DNA measurement is not in and of itself novel. But we had this insight into a way to do it, which we thought was pretty cool. Which that, aptamers are basically strands of DNA that can bind to proteins. And the standard way that you use, just to caricature a bit, the standard way that you use aptamers is that like, you take the aptamers and you mix them with a protein, and the aptamers bind to the protein. And then you like pull it down somehow, and you measure how many aptamers have bound to the protein. And the problem with this approach is that you get non-specific binding, which is to say that the aptamers are like not usually very specific in the first place. And so, you'll have this case where the aptamers bind to the target protein, but then they also bind to like this green protein over here. And so when there's not very much of your target protein, which is the case when you're looking at like these neurodegeneration markers, you end up getting like just detecting this background that's like green protein. You end up basically just detecting the background, and you have really low specificity. And so the way that the kind of insight that we had was that we could basically turn this problem around on its head by using this idea, which is called a strand displacement. So usually people think about strand displacement in the context of DNA, and what they think about is, if you have one strand of DNA hybridized to another one, then you have a longer a longer strand of DNA that comes along that can hybridize with like a like higher binding energy, lower binding energy, whatever, then the second strand can actually like knock the first strand off, basically, can displace the first strand off. And so that's, people think about that a lot in the context of, you know, various bioengineering applications. And what we said is the same principle should really apply to proteins. That if you take, if you have your aptamer attached to a surface and you have a complementary strand hybridized to that aptamer, and then you introduce your proteins, you have your both like this this specific protein in orange and this non-specific protein in green, that if you engineer the binding affinities correctly, the aptamer can bind to the target protein and can release this complementary strand. But the thing about this that's different from like the standard approach is that the aptamer is never going to give up the complementary strand in order to bind non-specifically to something. And so then the complementary strand gets freed in a way that's much more specific than than like this non-specific, you know, than just the aptamer binding to the protein. And then you can detect the complementary strand. You can do that very sensitively because it's DNA, right? And this lends itself to a really nice kind of assay format where you have, you know, a probe that has like some these aptamers on it, which are hybridized to their complementary strands. You stick that in a solution that consists of a protein that has your target protein in it. The target protein displaces the complementary strand. You take the probe out, and then you can just quantify the amount of complementary strand in solution. And this is really like extremely preliminary, but we had some kind of interesting results, which we're still trying to validate. But where, you know, we were able to, using this approach, we were able to detect spike protein, COVID spike protein, which we use just because we have it lying around in the lab, down to like 200 attomore, or maybe if you squint, kind of 40 attomore sensitivity, which is, you know, on the order of like dozens of copies of the protein. So this is really cool. We're following up on this, and definitely just like an example of the kinds of things that we're really interested in. And then the last thing that I really would like to say, we're running over, so I'm just going to keep it super short, is on the topic of viral vectors. Some of the projects that we're working on, we've become really interested in cell type specificity for viruses and for LNPs as a result, partly of these high-profile deaths that people may have heard about in the Astellas trial. Basically, some of the recent AAV clinical trials have seen deaths due to liver failure in patients. And the reason that's happening is because in order to get an effect in patients in systemic AAV delivery, you have to deliver extremely high titers of the virus. And that can just lead to liver failure as the liver tries to clear the virus from your system. And it has already been known that cell type specific viruses can lower the dose that's needed in order to achieve efficacy. So this is just a good example from Pardis Sabeti's lab at the Broad. Recently, they came up with a muscle specific AAV, which is here on the bottom, and just showing that, you know, at the same dose, you end up with way less AAV in the liver, which is what's happening at the top with AAV RH74, and way more AAV like in the actual muscles, which is happening at the bottom. And so then if you want to achieve the same clinical effect, you can lower the dose, which is then important for safety. And typically there are like two ways of going about increasing specificity. One of them is to come with a capsid variant to sort of do some like directed evolution or some high throughput screening to identify, which is like this column on the left from Ben Deverman's paper about CREATE, to identify viruses that can specifically infect one cell type. The other way that people do it is to come up with enhancers that are very specific to a particular cell type, so that the AAV will infect cells broadly, but will only be expressed in one cell type. That actually like doesn't solve the problem that we were just talking about, because you still need super high titers. And in both cases, you basically end up in this situation where you need to like come up with a new AAV for every cell type that you want to infect. And so the way that we think about this, it's a little bit like we're in the zinc finger and tau effector kind of era, where every time you want to target a new region of DNA, you need to go and create another protein that is specific to that region of DNA. And so then all I can say about this is that we're working on basically coming up with a programmable method for generating cell type specific nucleic acid delivery. Yeah, that's about it. And so again, if any of these projects are exciting to you, please get in touch. We would love to, I'd love to chat. All of these projects are, 3 of the projects I talked about are funded recently, like in the past few weeks. And so I'm hiring rigorously, or hiring rapidly. And so yeah, would love to chat if anyone's interested. And I'd just like to acknowledge also all the people who I've worked with on these projects. So these are people from this is not even the right acknowledgement slide, so messed that up. But my many funders, and then also Daniele Sarvatini, who is postdoc in my lab working on the viral vector project, Patrick Arnott, who's working on the diagnostics project. The ticker tape people or timestamps people are listed here. And then also on Connectomics, a bunch of people, Bobayan in particular, Jürgen Kornfeld, who's at Max Planck, who has been a great collaborator on that, Ed Boyden, who's been a great collaborator on that, Michael Fee, also at MIT, who's been a great collaborator on that. More people, but yeah, that's about it. Well, we can leave it there.
Thanks, Sam. Do you want to unshare so we can see your face again?
Indeed, yeah. Stop sharing.
I have a feeling we might want to share again in case people have got specific questions, and or you use your slides to do it. So we might be backwards and forwards on this.
Sure.
It was great getting all that in. Lots to get in, and another thing.
Marathon. So.
Yeah. So everyone who's listening, we now have about 10 minutes or so for formal questions. Either raise your hand with the raise hand icon on Teams if you want to speak, or write it in the text box. There's already one in the text box to get us going from Satya Prakash. Satya, you can unmute and put your camera on if you want to ask the question yourself.
Okay.
Yeah, no pressure.
Okay, so anyway, okay. So hi Sam, very nice talk, and thanks for Tom as well to organize. So I'm asking, this is possible that you talk about aptamer to diagnose the protein. So these aptamer can diagnose the endogenous expression, yeah, endogenous protein. So does it possible?
If what you mean is can it be used to like diagnose diseases based on endogenous protein expression, like certainly that's the goal.
Yeah,
yeah, certainly.
Okay, thank you. Yeah.
Okay,
thank you. Yeah, because we are also planning something on the mRNA based endogenous mRNA expression diagnosis.
Yeah.
So okay. Thank you very much.
Yeah, thank you.
All right, we have a question from Mr. Rowlands, Dr. Rowlands. Chris, do you want to ask it in person? You're going to make me read what you wrote.
I was going to make you read it, but yeah. Yeah, no, I can't remember whether I asked this to you previously, but regarding the ticker tape work, what other effects that exogenous effects affect the tick rate, like the clock rate? Because presumably, changing the rate at which the enzyme alters the DNA will change the affected or the apparent time that's elapsed since the RNA was synthesized. Sorry, RNA, not DNA. So things like temperature, light exposure, expression of proteases, expression of your modifying protein. I've forgotten the name of it.
Yeah, well, so it's a great question. The temperature, light exposure, I mean, it's in cells, and they're like at, they should ideally, I'm sure the temperature would affect it, but they should ideally be at a constant temperature because they're alive. As for, I think that the best way to answer this question is just to say that it's probably cell type specific, which is a pain in the ass. And certainly, like, there needs to be some calibration. If you want to get absolute timing out, there needs to be some calibration. Usually what you're just interested in is how old, or like, how old are RNAs in condition A compared to condition B, right? And so for that kind of thing, you don't really need a calibration. But otherwise, yeah, if you want like absolute timing, then you have to worry about all this stuff.
Chris?
Let me know if that wasn't a satisfactory answer.
Sorry, I just need to unmute again. Thanks. Awesome.
Thank you.
I'm going to jump in with a question whilst we wait for more from other people. So I really liked your analogy of the connectome and the genome.
Yeah.
Right? So Human Genome Project took ages, cost an absolute fortune, and they only sequenced one genome. It's kind of a bit of a hybrid mismatch of a bunch of people's genomes, although Craig Venter sequenced his as well, right?
Yeah.
And a comment on as criticism on the Human Genome Project, which sounds great in retrospect, but is ridiculous if you think about it at the time, from George Church, was that we shouldn't be putting all this money in to sequence one genome for $3 billion. We should instead find a way to sequence 3 billion genomes for $1 each. Right?
Yeah.
He said that last week at a meeting I was at, which is obviously ridiculous, because you can't do both.
That is one of his lines. That is one of his lines. Yeah.
So Connectome, right? So there's a lot of money gone in this, and what you presented struck me as very expensive work, like in terms of materials and stuff. So how can we ever get, Connectome kind of research down to what we're doing in genomics with Illumina and nanopore scale?
Oh yeah, I think it's not that. I think the expensive part of what we're doing is the antibodies, and certainly coming up with the
and the microscopes.
And well, okay, but like the Illumina, like a sequencer is also a microscope, right? So obviously,
I bet it's cheaper than the microscope you're using. Maybe.
It is definitely cheaper than the microscope that we're using. That's right. But I think that the thing that puts people, but a NovaSeq is more expensive than the microscope that we're using, right? Like the capital cost for a NovaSeq is way more expensive. So I think that the capital costs, I tend not to worry about. And in terms of consumables, unless they're outrageous, I tend not to worry about them, but I don't think the capital costs are outrageous. And then in terms of consumables, I think that it can probably, it scales once you're done, once you figure out what you're doing. I mean, the issue with that kind of project is that when you screen through 200 antibodies at, 500 pounds an antibody, it's like expensive, right? But I don't think that there's, I mean, I actually think of this method as like likely being a way to accelerate and bring down the cost of connectomics. Because I think the thing that will kill connectomics from a cost perspective is this phenomenon where you have to go through and like image the entire brain every time you want to see anything. Like what you want, like most experiments that people are going to want to do in connectomics are going to be to look at connections between region A and region B, like two specific regions. And like if they can just image region A and then also image region B, that is going to be cheap. Whereas imaging the entire brain just to answer some question about A and B is impossible, right? So I think that's where you get the savings. It's in the volume as opposed to in the reagents.
Let's see. There's another
Chris Rowland's question.
Come on, Chris.
What can I say? It's all fascinating. Cool. Oh, hang on. Have I lost you? There we go. I was interested in your kind of rainbow on steroids protein expression. One of the advantages of rainbow is obviously you're only encoding 3 proteins, so getting the relative expression levels between the 3 of them is not trivial, but achievable. But when you're talking about 30 proteins all being expressed, like what makes you think you can control the expression levels sufficiently well that you're not going to either massively overexpress one and fail to express another one, or tax the cell production, the protein production machinery of the cell, so that the one at the end doesn't get expressed to anywhere near the same levels as the one at the beginning of the promoter, or whatever?
Yeah, that's a great question. The answer is just that mouse light. The way that mouse light works is that they they deliver like 30 copies of GFP per cell, and then they also deliver like so they have like super high titer GFP and super low titer Cre to ensure that their neurons are like absurdly bright and very sparse. And so that was what convinced us that we can express like 30 things in a cell without killing it. Now, as for the rest of the other parts of the question, reliably switching them on and off. In fact, we're just using stochasticity of viral infection. So it's like, you titer the virus so that every neuron gets infected with like MOI 5 or something, or MOI. Ideally you want, like, if you're 30, you want MOI 15, but that's like hard to achieve. So, if you have 30, you maybe go for MOI 6 or 7 or something like that. Sure. And then what makes us think that they'll be expressed like at similar rates? We just observe it.
Cool. Thank you.
Yeah.
Okay, we've hit 5 o'clock now, so the formal bit is over. I think the recording